Hey, what is going on? It has been a while, but it is Friday, March 25th, 2022, and we are almost at the end of Metabolism March. I did skip a few days, so I might have to shunt Metabolism March or try to stuff a little bit of it into Ascension April. I don't know. I haven't thought of a term. Amine April, amino acid April. I don't know. Something that qualifies April in the way that I want to in terms of our life extension studies. So let's get right to it, deep into the thick of things. It's Friday night, so we want to talk about lactose today. Okay, what is lactose? So you all know what lactose is. Lactose, or a lot of you know what lactose is, even those of you who are not chemically minded amongst us. Lactose is a disaccharide. Disaccharide is two sugars connected together. And what two sugars are those? Well, those are glucose here and galactose. Now, glucose and galactose. Glucose and galactose. What are glucose and galactose? What is the difference? Glucose, remember, has a specific orientation of the hydroxyl groups around its main carbon ring, primary carbon ring, and galactose has a different orientation of hydroxyl groups around its main carbon ring. Okay, so what's the difference? How does it work out? So here, this is actually the best way to see the difference and how does lactose the whole tie in is that glucose has a OH group facing up, the hydroxyl group facing up, so you can think glucose kicking up, and galactose's hydroxyl group is on the 5 prime carbon, is 6 prime carbon is facing down, right? So OH is facing this way, and in this particular case, because this OH faces down, the glucose can bond through an O-glycosidic bond, more specifically known as a 1,4 beta, a 1,4 beta glycosidic bond, O-glycosidic bond. So, and again, sorry, I needed to clarify. It's the one carbon of lactose combined with the of glucose combined with the 4 carbon of galactose, right? So, and that's how this bond forms. And so, lactose, as you know, or as many people know, is a large molecule and it can't be uptaken by your cells. Glucose specifically, can be transported into your cell via GLUT transporters and then converted to glucose 6 phosphate, and then afterwards go through the whole um, glycolysis or glucogenesis pathways or whatever other pathways, because that glucose 6 phosphate branches into different pathways, right? And galactose itself, now uh, galactose, if it's in the cells, needs to, or wherever it is, it needs to be converted by itself. Remember, galactose by itself needs to be converted into glucose, and there's a glucose galactose interconversion pathway which allows this to happen. Okay, and so since just also to note that galactose and glucose are actually what's known as epimers, right? Epimers are basically, they are they're compounds that are mostly the same except for the orientation of some of their side groups, like this hydroxyl group here and here around a chiral carbon. Okay, so that's an epimer, right? So an epimer is basically an isomer, but it's, again, it has a different stereochemistry. It's an isomer with different stereochemistry. Okay, so but when the two isomers of the different stereochemistry are connected together, glucose and galactose, you get lactose. Sounds like you're combining a lot and you're getting less just by the naming convention, but I'm sure there's some reason for that. Anyway, so lactose itself can't make it into your cells. So what happens is lactose accumulates. Where does lactose come from? Well, lactose comes from meat and cheese. Okay, and it comes from milk, cheese, dairy stuff, several different places that many people know about. And the inability of the cells to uptake it requires there be a breakdown process for this lactose. So normally you have an enzyme, or you have many enzymes, which break down this lactose, and the enzyme that primarily does this is called lactase. But some people who are lactose intolerant, including myself, do not have lactase. Okay, so let's just take lactase. Right, if you don't have lactase, images. Right, lactase is lactase chem structure, right? So we can find an enzymatic structure. So lactase is this beautiful thing here, which through the specific biology we won't go into, but it involves water, right? The usage of water molecules, it will cleave lactose into glucose and galactose, right? And so this is kind of the process here, using the H2O water binding site. So again, we'll go into the details of more specifically how this happens. A little precursor to that is this substrate enzyme complex diagram, which basically requires that both galactose and glucose fit into this active site of the enzyme, and then water comes by, and the amino acid side chains will split galactose and glucose apart when water is involved. Okay, so that's at a high level. That now, what happens, right? How does one thing we should talk about is what happens in the galactose pathway? Okay, because the metabolism of galactose, right, which occurs in this occurs in the liver. Okay, remember, galactose doesn't make it into cells the same way glucose does. So galactose has to be converted to glucose, right, or glucose 6-phosphate, so such that it can be taken in, and it can be used as a reagent in glycolysis or glucogenesis, because once you get glucose 6-phosphate, you can use that reagent. Lactose, remember, lactose broken down into galactose and glucose. Galactose now is by itself and needs to be converted into glucose, essentially. So that's where galactose metabolism comes into play, right? And galactose essentially has this chain of enzymes here, galactokinase, which is the first one. You get the result is galactose 1-phosphate, but remember, we're looking for glucose 6-phosphate. So galactose 1-phosphate now has an interesting way of getting to glucose 6-phosphate. Okay, so first, galactose 1-phosphate has to convert to glucose 1-phosphate, and it's done through a intermediate enzyme which is a mouthful, called galactose 1-phosphate uridyl transferase. Okay, so what happens with galactose 1-phosphate uridyl transferase? Okay, so a molecule called UDP glucose comes by. Okay, UDP glucose comes by. Galactose 1-phosphate uridyl transferase essentially takes the UDP, which is uridine diphosphate, takes it off of the UDP glucose, and puts it on the galactose, and thus creates UDP galactose. Okay, so then what happens? This glucose 1 is phosphate, is what you essentially have as a result. And then this glucose 1-phosphate needs one extra step just to make sure it's in the right configuration such that it can go into glycolysis. So glucose 1-phosphate is interacted upon by an enzyme called glucose 1 mutase, which essentially transfers the phosphoryl group. Okay, so again, glucose 1 mutase involved action. We can look roughly glucose mutase, we can look quickly at what's going on. And some of these enzymes is interesting. So phosphoglucose mutase is an example of what glucose. What's the reverse reaction? A conversion of glucose 6-phosphate into glucose 1-phosphate, right? And that has its other implications in other cycles that we've talked about. But in this cycle, we have the reverse reaction, right? So essentially, it repositions the phosphate group from the 1 to the 6 in what we need. Okay, so once we have um, glucose 6-phosphate, we're hunky dory. So essentially, there's other enzymes here. We talked about a UDP galactose. Now, how does UDP galactose go back to UDP glucose? Okay, so that's a whole other thing. We're not going to get a ton into that, but you were wondering, where does the UDP galactose and UDP glucose come from? So UDP galactose is what's left, right? It's a byproduct of the left chain, right, of the conversion of glucose UDP, the conversion of galactose to glucose 6-phosphate for glycolysis. But the byproduct UDP galactose now has to be recycled such that it can be used in the galactose to glycolysis pathway that we've already talked about. So essentially, the epimerase comes into play again. And remember, the epimerase, UDP galactose. Okay, think about it. UDP galactose and UDP glucose, they sound completely different, but they're very close to each other, and therefore, an enzyme we've already done an epimerization reaction earlier. We only need to do one more epimerization reaction, or set of one more epimerase reaction, such that we can move the phosphate again to create UDP glucose. And then UDP glucose is again the reagent that restarts the cycle on the left. Okay, so that's a lot. Now, we can talk at length, continue to talk at length about this, and I would like to mention essentially diseases that are a result of issues with lactose and lactic acid pathway, galactose pathways. So much lact going on. The glucose galactose interconversion pathways. One of them is the simplest, lowest yield one is lactose intolerance, which means you just don't have the enzyme lactase in quantities sufficiently in your digestive tract, such that the gut bacteria, right? You have gut bacteria that require this, that require these inputs, these sugar inputs to function. So essentially, what happens with lactose intolerance present, the lactose won't be broken down. The gut bacteria will consume the lactose, and therefore there will be a buildup of gas that occurs, H2 gas and a few other kinds of gases, but primarily H2, which causes bloating and indigestion. Okay, so and then we can see the issues with the pain of the pain tolerance, right? Or lactose intolerance. We see what issues it causes, right? And so that's here as we described. Now, let's talk more about, there are two other diseases which are worse than simple lactose intolerance, and those are autosomal recessive disorders, and those involve, that disease is called galactosemia. Okay, and that's when galactosemia essentially occurs. It's a disease. First, let's talk about what its issues are, what its symptoms are. And there's brain damage, cataracts, jaundice, enlarged liver, kidney damage. And so basically, if a galactosemic infant is given unmetabolized milk, sugars build up, damage the liver, eyes, kidney, and brain. So galactose, why is that the case? How could that happen? So basically, remember, there's an enzyme called galactokinase, which does the conversion of galactose to glucose and allows it to is involved in that process. So essentially, when galactokinase, due to the presence of this disease, of this genetic knockout on your on two of the alleles that are coding for galactokinase are present, right? Then galactokinase isn't present anymore, and galactose then gets acted upon by an enzyme called aldose reductase. Aldose reductase, right? Aldose reductase is the first catalytic step in the NADPH-dependent conversion of glucose to sorbitol, so that's involved in other pathways, but it's important in our case to know that aldose reductase then converts galactose to galactitol, okay? And once galactitol is present, galactitol in high quantities is what causes galactemia, right? And then there's different levels of galactemia, and we should know that this is a serious disease because of its implications, permanent implications for children, and there was even a worse disease, and that's not when galactokinase is removed. That's when the enzyme galactose 1-phosphate uridyl transferase is unencoded for, is knocked out. And when that happens again, you're unable to send glucose to the galactose to the glucose pathway, and thus you have a buildup of the byproducts of this reaction, and thus you cause galactosemia, of a much worse version called classic galactosemia. So both genetic disorders are pretty bad. And thank your lucky stars if you only have lactose intolerance. So that's a lot. I mentioned a ton of enzymes here. I kind of just wanted to put galactose in the picture because galactose is important, and because it's present and it's involved in metabolism, and it eventually aids in glucose metabolism through glycolysis. But yeah, I didn't go too much into the chemical structures of these enzymes, but at some point in time, I will talk about all of these enzymes in an enzymatic deep dive at some point. Okay, so if you like this video, like, subscribe. I'm sorry I took such a break before, but I'm back, and let's talk again soon. All right, bye-bye.